Hi, everybody. So today we'll be talking about deep learning and its applications in biomedical research, especially in single cell sequencing. So, deep learning is actually, a subset of machine learning, which in turn is a subset of artificial intelligence. So I believe Sandeep has already covered a lot on machine learning, and many of the principles that he covered will still apply to deep learning as well. So many of the things that you have learned, you can also apply those things to deep learning. So the deep learning relies on neural networks. So we will look more into what neural networks are and their structures later. So the major difference between machine learning and deep learning is that for machine learning, when you have the input, you need to extract the features that you want to use for predictions. And so you sometimes you have to do like feature engineering as well. Say you have two columns. You also generate a new column by multiply those two columns, for example. But in contrast, for deep learning, usually you don't do much feature engineering. So when you have input, you just feed the data into neural networks, and the neural networks will generate output for you. So, for example, if you have an image, you just put image, feed the image into the neural network structure, like convolution, send a name, and then generate output for you. So that's the major difference between this machine learning and deep learning, in my opinion. But other than that, the underlying many of the underlying principles are the same, or very similar. So we heard a lot about deep learning because it's applied, it is being used for many aspects of our lives. So like image classification, and also for entertainment, Amazon used deep learning to recommend the books for you. Netflix used deep learning to recommend movies or TV shows for you. And it is also being used for like facial recognition, and also for auto self-driving cars, like Tesla. They all use deep learning. So today we'll be focused we will focus on the applications of deep learning in medicine and biology. We have heard scientists use deep learning to detect cancer, or other diseases. So we will dive deeper into this. So in terms of the applications of deep learning in biomedical research, I think there are two major types that are being used for the research. One is supervised deep learning, and the other one is unsupervised deep learning. So for supervised deep learning, the basic idea is that you have labeled samples. So in this example, we have pictures that are labeled with either cat or dog. And then we feed those images into a neural network, and then the neural network will tell us, whether this, picture is a cat or is a dog. But that's the basic idea, right? So you have labeled samples, and then you want to classify, new samples to either to one of those categories. So this is a side project that I worked on before. So basically, we have X-ray chest X-ray images. So so based on those images, we want to know whether a person has pneumonia or not, right? So so we use those images and then use a neural network to tell whether this X-ray image is from a pneumonia patient or not. So using very simple techniques and the very preliminary result shows that we can reach accuracy of over 91%. With very little work. Maybe you can from the beginning to the end, it only takes about 1 hour or 2 hours to get this result. So this really shows the power of deep learning. So even though I have a background in biology, I don't know how to read those images. I cannot tell whether a picture shows pneumonia or not. But this neural network structure can do that with a over 91% accuracy. So I think that's pretty amazing and shows that the potential of deep learning in biomedical research. So you can read more on this in my blog post, and I also shared the code in GitHub. So you may want to take a look if you are interested in this, using deep learning for analyzing biomedical images. This not only applies to X-ray. You may also apply this to other types of images as well, such as microscopy images. So the other aspect is unsupervised deep learning, right? So the supervised and the unsupervised idea is also the same for machine learning as well. So in unsupervised deep learning, we don't so each sample, we don't have labels for each sample. So we don't know whether it's a cat or dog. We just have this sample or image. Right. So for these types of data, we want to do clustering, right? So we want to find samples that are similar to each other, right? We can cluster them to form different clusters, or the cyan cluster or green cluster, which are within the clusters, the data points or samples are very similar to each other. We can for clustering and the other applications for dimensionality reduction, right? So you can imagine that for in biomedical research, so if we do sequencing, right, we will have thousands of genes which can be treated as features. So we have thousands of features, but say if we want to visualize the data, how can we cannot visualize more than like 3 dimensions, right? So we want to project our data to a 2 or 3 dimensional space in order to visualize the data. So that's where deep learning can come in to help us visualize high dimensional data. Although we have traditional methods like a PCA, which can do linear dimensionality reduction. But the power of deep learning or neural networks is that they can do nonlinear dimensionality reduction, so they can keep information that may be lost by using methods like a PCA. So in this example, we have data with 3 different features, and then we can reduce the features, reduce them to a 2-dimensional space, and then visualize them. So again, the idea is that even though we project our data to 2-dimensional space and reduce the dimensions, but we still keep the global structures, right? So if the data points are different in a high dimensional space, we still want to separate them in the 2-dimensional space. We can still say the orange dots are different from the green dots, for example. So really, so in today's talk, we will be focused on the unsupervised deep learning side. So one major application of unsupervised learning is for sequencing data, right? Like I just said, we have thousands of genes, and we want to cluster say the cells and visualize them in a two-dimensional space. So today we'll be focused on single cell sequencing, right? So we may, like a few years ago, before the 10x technologies become, so popular as of today, the bulk sequencing was more commonly used, right? So we take a sample, we have like maybe a few thousand cells or even millions of cells, and then we sequenced the mixture. So at the end of the day, we gather like an average expression across all the cells, right? We measure the gene expression in thousands of cells, but we cannot tell, the difference or heterogeneity within this cell population. All we got is an average output, which is not ideal, right? Because we know that each cell is different. If we just look at the average, we wouldn't be able to tell, the heterogeneity within this cell population. So that's where single cell sequencing shines, right? So we can sequence the transcriptomes, of each individual cell, and then we can really tell, we can really identify, cells with different gene expression profiles, and even identify rare cell populations. Say we may have like 10 cells out of say 1,000 cells or 10,000 cells. Now with single cell technologies, we can really identify those cells. So maybe those cells are very important in terms of the generation of certain diseases, right? So they may drive the pathology, or they are very protective, right? They can protect us from certain diseases. So single cell sequencing really gives us the power, to identify the heterogeneity of our cells, basically. So for these types of data, you can imagine that each cell is like a sample, right? And each gene is a feature. So now if we sequence like thousands of cells, right? Now we have, say we have the samples as rows and the features as columns, right? Now we have a gigantic table with thousands of rows and a few thousand of columns, right? So now single cell sequencing really generates like big data, right? So which give us as a biomedical or bioinformaticians, right? We have the data to work with, which we can use deep learning to analyze the data. So today we'll be looking at clustering and dimensionality reduction, right? So how do we use deep learning to accomplish those goals, right? So like I just said, we have genes as columns. Here we look in this picture, they put genes as rows, cells as columns, but the idea is the same. So we have this data, right? So how can we condense this data into a two-dimensional space, right? So we still have this many of cells, but imagine that we only have 2 rows, right, in order to visualize them. So what we do is that we use a okay. So we use a deep neural network architecture called autoencoders, right? So I encourage you to read more about autoencoders. It has many variations. There is also a neural network architecture called variational autoencoders, right? So there are many different kinds of them. So they're very useful, and I encourage you to read more about autoencoders. Anyways, so now we use fully connected layers. So we reduce. So this is here we're looking at one cell, right? You can imagine the green circles are the genes, right? Then we, use those neural network layers to reduce the number of features so say first we have like 2,000, and then we reduce them to like 100, and then reduce them to like maybe 20, and then to 2, right? Or to how many neurons you want. So, and then we put, after, encoding the data to a lower dimensional space, which we call, bottleneck layer. And the goal is to, reconstruct the data, the input from this condensed, compressed data. So the idea is that, we learn the essential information from the input, and then we use that essential information to reconstruct the entire input. So in this process, we got the most important information from the input, and then we can use the most important information for visualization or for clustering, right? So that's the basic idea. So this is the general overview of autoencoders. So we compress the data and then learn the most the essential information within the data, and then reconstruct the input from the compressed data. So that's the basic idea. So we don't always compress the data into a two-dimensional space, because if you imagine that it is really hard to learn all the information, right, in just 2 neurons. So that's very difficult. So usually we use more than that. Usually I use 20 or 32, something like that for the bottleneck layer. Because if we have more neurons, we can learn more information from the input. So in today's example, we're going to be looking at cite-seq, which is a type of single-cell sequencing technology. So in normal single-cell RNA sequencing, we only look at the transcriptome, right? We look at the gene expressions of a cell. But the power of cite-seq is that it also measures the expression of surface proteins. So cite-seq gave us the output of cite-seq is the gene expression and the protein expression from the same cell. So that's really powerful, because by just looking at gene expression, you only get like one aspect of a cell, right? So protein expression also give us another aspect of a single cell. So if we combine those two data modalities, the hope is that we can generate more insights into a cell. We got a more comprehensive profile of each individual cell. So for example, the gene expression, the expression of certain genes does not correlate with the protein expression of certain molecule. So if we combine those two, we may get a big picture, better picture of the profile of our cell. So that's the idea. So basically combine two data modalities for each for single cells. So just similar to RNA sequencing, right? We also get so for RNA sequencing, we got a count matrix for gene expression, right? So here I put genes as columns, cells as rows, right? We got the counts for each gene, each cell. So for cite-seq, we have 2 metrics, right? So one for gene expression as usual, but we got another count matrix for proteins, right? So again, in the rows we have cells. In the columns, we have proteins, and we got accounts for each protein in each individual cell. And the cells are the same for each, or the rows are the same for the 2 matrix, right? So we have the information for both gene and protein for each for the same cell. So that's the idea. So we so here we got an additional challenge, right? So is how can we combine these two, right? So if we just concatenate those two, you can imagine that for genes we usually have thousands of genes, but for proteins we usually have maybe a few dozens of proteins. If you just concatenate those two, the protein, the information from the proteins will just be lost, because we have so many genes, and the gene expression data will dominate our downstream analysis. So the challenge is that how can we better integrate these two data modalities. So in today's coding example, we will be looking at one a simple strategy to integrate those two data modalities, right? So we have gene expression as shown in green, and we have protein as shown in orange. So the idea is that we first compress them separately. We have one encoder for gene and one encoder for protein. So we can compress thousands of genes to, like, say 80. And then we compress the proteins to, using, like, say 20 neurons. Now they become more comparable, right? We have 80 neurons for genes and 20 neurons for proteins. Now they become more comparable. So we make sure that the protein data is not lost, or overshadowed by thousands of genes, right? So then we'll concatenate the output from the first encoders, and then we use another encoder to compress the concatenated data into a lower dimensional space, and then we try to reconstruct both the gene and the protein expression from the compressed data. So we'll be looking more at this in our coding example today. Yeah, so today we'll be implementing the neural network or autoencoders for cite-seq using PyTorch. So PyTorch is a deep learning framework in Python. So you can check out the code in my GitHub repo, and I also wrote a blog post on this. You can read this as well if you are interested. Yeah, this may serve as a resource for you. So if you are working on single cell sequencing data, or you are interested in this, there are many packages available for single cell analysis. So here I listed probably the three most popular packages for single cell analysis. One is Seurat. So Seurat is written in R. So if you use R, Seurat may be your go-to package for single cell analysis. The other two are in Python. So ScanPy is very similar to Seurat, but it's in Python. SCVI, the third one is a deep learning based single cell analysis package. So it is also written in PyTorch. So if you are interested in deep learning, you may want to check out SCVI. It's a really cool tool. So the idea of SCVI is very similar to what we will cover today. What we will cover today, but it has more advanced features, and it use more complex models. So you may want to check out SCVI out. But the idea, the basic idea is the same. So today, hopefully today's session will also help you understand, what SCVI does under the hood. All right. So before we looking at the code, do you guys have any questions regarding the presentation?
Yes, sir. I have a question.
Sure.
Sir, do we need all the data just for expression data and protein data also for this analysis?
Sorry, can you repeat that?
Sir, do we need both type of data matrices, expression data matrix and protein data also?
Yeah, so no, you don't have to have both. So normally people only have, say, RNA data, right? So you can still use the code for RNA sequencing data only. You don't have to need proteins. So yeah, you just need to have. So today we'll be looking at both RNA and the protein, but in reality, if you just have RNA, it works too. But it's rare that people only have proteins. Normally, they would have.
Yeah, that's why I'm
RNA.
confusing, because we usually have RNA-seq data, complete RNA data.
Yeah, if you just have RNA, you can just use the. It's fine. You don't have to have proteins.
Oh, okay, thank you, sir.
Sure. Any other questions?
Excuse me, sir.
Sure.
Is the package RAM intensive? Can it be worked upon in your personal laptop? PyTorch?
Depends on, yeah. So what laptop do you have? Do you have a GPU?
No. Actually, it's a 8 GB RAM and AMD Ryzen 5 processor.
Okay, yeah. So you have so for PyTorch, you can use it in CPU. So you don't have to NVIDIA GPU for PyTorch, but for our applications, we have the tabular data, right? We have like count matrix. It doesn't make a big difference if you just use CPU, but if you are working on images, it's it would be very slow if you just use CPU. Say, if GPU trains like one epoch in say 5 minutes, you may train like for hours using a CPU if you don't have an NVIDIA GPU. So
no, sir.
yeah.
Actually, I was analyzing single cell data recently, and I faced a problem with the Surate package. The new that integrate anchors package is always running out of RAM every time I try to integrate. So I asked for curiosity if PyTorch can be worked upon in a low RAM the conditions or anything like that, because single cell data requires too much RAM and it's very RAM intensive in
So you're
personal
talking
computer.
about RAM?
Yes, RAM.
Okay, so you're not talking about GPU. Yeah, so I don't know if you don't have enough RAM. Because do you have like HPC infrastructure at your institute?
No, actually.
You don't? Yeah, so for us, we just I don't do like any computation on my laptop. I use our HPC. But you may want to check out one of those cloud platforms like AWS or Google platform, to use their clusters. Yeah, it's very challenging to do like a heavy computation on your laptop. You're limited by the resources, so you may want to check out one of those cloud platforms. Yeah. So some of them have promotions. If they do, you may want to check it out and try it out, because I think I used Google cloud platform once. They give you like $250 as a promotion, so you can use it for like quite a long time. $250 is a lot. Alrighty. So if we don't have further questions, we're gonna go on to look at the code. All right. Yeah. So before we look at the code, I just want to show you the PyTorch website. So it's really cool. It shows you how to install PyTorch. So whether you have a Mac, Windows, or Linux, you can install PyTorch using either Conda or pip. And like I said, you can just install a CPU version if you don't have a NVIDIA GPU. Unfortunately, AMD doesn't work. You have to use a NVIDIA card. So you can use CUDA stands for GPU, and CPU version if you don't have a GPU, and then you just copy paste the command to install it. And then they have beautiful tutorials. I really encourage you to check it out if you're interested. They cover from the basics, to tensors, what are tensors, data sets, how to build like a customized data set or data loader in order to train your neural network. And then you can look at the certain more specific applications like for images, audios, or text. The tutorials are really cool, so you may want to check them out if you're interested. Okay, that's for PyTorch. So yeah, so these are the notebook for our encoder analysis for cite-seq. So I'm just gonna import all the packages that we will need for this. So I'm running this as we go, so it may take some time. So then we will, read the data. So by the way, the data is also included in the GitHub repo. So if you wanna, try it out yourself, just clone the repo and run it on your own laptop or on the cloud. So yeah. So now we read the RNA data. It's just like a CSV file. And then so we use pandas. Then you can just take a look at the structure of the data. This may take a second because it's quite large. Yeah, just ask me questions if you have any. You don't have to wait till the end. Just interrupt me at any time if something's not clear. Yeah, now we have the data, right? So we have, in each row, each row is a cell, and each column is a gene or feature, right? So now we have this count matrix for RNA, and we do the same for proteins, right? Now, proteins we have much fewer proteins, so this is much quicker than RNA. Like each column is a protein, and each cell, or each column, or each row is a cell. And then now we can say, how many genes we have and how many proteins we have, right? So we have 2,000 genes and 25 proteins. And then we can, concat those two tables to form a single data frame that we can work with, right? So just similar to machine learning, right? So we have a trained data set and a validation set. So we just split those the data set into training and validation using scikit-learn. So now we have about 27,000 cells for training and about 3,000 cells for validation. And this is just looking at one example, right? This is the first cell. We have the numbers, for genes and proteins. So for PyTorch, for training a model in PyTorch, right? You need to feed the data into the neural network. So in order to do that, you first need to have a data set. Data set basically contains all the data you have. So we can look at the code. So here we're using a customized data set, right? So for our data. So here's the code, right? So for custom data set, you just inherit from the data set class from PyTorch, and you need to implement 3 functions, right? The first is init. You basically say, oh, my data. You just initiate with your data. So in our case, our data is a NumPy array, right? So the dimensions, number of genes times number of features. And then you need to tell, how many samples you have, right? Basically, it's the length of our data. And then you need to tell it how to retrieve one example from your data set, right? So basically, for NumPy, because so each sample is a row. So we just, index a row, right? Just if we have an index, we just extract the index, using the index from the data. So this is very simple. So to return, you basically return your input and target, right? So in our case, because we're trying to reconstruct the input, so the input and the target are the same. They're both, the cell, right? The row, basically. So that's it. And that's the data set. So we use our train and validation to build our 2 data sets. Then we need to build our data loaders. So with our data loaders, so for training your network, you don't train like with one example at a time, right? Usually we train with batches. So in batches, like, so you can specify your batch size, like here. So here we're looking at a batch size of 64. So we train 64 cells at the same time as a mini batch, as a batch, right? So we first build the data set, and then we tell, we need a data loader which loads 64 cells at a time from the data set. So that's the idea. So for train, we also shuffle the data, so because if you just train with the same data every time, the model may learn, like, so basically you want to avoid overfitting, right, to your train data. So you want to shuffle the data for train, but we don't shuffle for validation because, there's no really purpose, for shuffling the validation data. So we have the data set, and then we build the data loader to know, 64 samples at a time to fit into our neural network. So here we're looking at like one batch, like, so we have train data loader, and then we grab a batch of it. So now we can, so if everything is correct, we would have, for X and Y, the shape, how do you say, the rows would be 64, right? So we can check that. Indeed, so we have 64 samples, and these are our features. So we have 2,000 genes and 25 proteins. So it's 25. So this is as expected, and then we can build the model. So basically, this is the the model I just showed you in that presentation, right? We have encoder for RNA and an encoder for protein, and then we give it like a bunch of parameters, and then we can get the model. So the model is implemented in the model.py file, and so we will quickly go through this together. But you can always go back and then check it out on your own if something is not clear. So for autoencoders, right, we have an encoder which compress the data, and then a decoder to reconstruct the input. So that's what that's how we would go about implementing this. So for encoder, we have we need to know how many genes we have, how many proteins we have. So I think one of you just asked the question, right? So what if we just have RNA data? Right? You basically said, for in our model, I consider this. So you can basically set, number of proteins, number of features for proteins to 0, and then the model will run, just fine. So now we specify, the encoder for RNA as we just showed, and then we have encoder for protein as well. So the parameters we give is the hidden RNA and hidden protein, right? So just meaning how many neurons do you want to compress the RNA to, and how many neurons do you want to compress the protein to. So we need to give those two parameters. And then the mean batch norm drop is basically how to say, abstract for 3 different layers, right? We do the linear layer, and then we do activation. Activation is nonlinear, right? So that's how we overcome the linear transformation methods, such as PCA. We use nonlinear activations. So here we use ReLU or leaky ReLU. It's basically, if a cell is positive, it's going to be the same, right? So it's but if cell is negative, we would just treat it as 0. For leaky ReLU, it has a small slope, so it's not completely 0, but it's very close to 0. And that's the idea. And then we do batch norm. So those are details that we don't have time to cover today, but you can check out the code and research on your own. So then after we have the encoders for RNA and encoder for protein, then we build encoder for the concatenate data, right? So we first compress them each individually and then concatenate them together, and we need another encoder for the concatenated data. So here we have another parameter called ZDM, which basically it tells the neural network how many neurons we want to use for the bottleneck layer, right? So that's it. So we then tell the neural network how to treat process the input in the forward function. So for if we have cite-seq, we can do the encoder RNA. This is the number of, so we slice the input to only have the protein, the RNA data, which is not for RNA, and we also slice the input to contain only the protein data, and then we run that through our protein encoder, and then we'll concatenate those two. But say if you only have RNA, you can just encode the RNA. Say if you only have protein, you can just concatenate, you just encode the protein. And finally, we run the compressed data to another to this final encoder to get our compressed data. So DEcoder just does the opposite. It just reconstruct the data from the compressed data. And then we can combine those two parts together in our autoencoder model. So we have an encoder and we have a decoder, right? We just run the data through encoder and get output, and then we further run the output from decoder to get our final output. So that's the basic structure of our model implemented in PyTorch. If something is not clear, just ask me or check out the code later. So now we get a model, right? So here we give it like hidden dimension. We use 85 neurons to compress the RNA, and we use 15 neurons to compress. 85 neurons to compress further compressed RNA data, and 15 neurons for protein. And then we concatenate those two, so we have 100 neurons, and then we further compress that 100 to a final size of 20. So at the end of the day, we compressed our over 2,000 features into 20-dimensional space. And then we have model. This is our model. We have the encoder, and then we have the decoder. So just like we just went through, and we can run a batch from the through the model, because we're like reconstruct the input, so the output should be the same as the input, right? So it should be 64 times 2025, right? and exactly as what we expected. We have 64 samples, and the output is 64 by 2025. So that's all fine. And then we need to train the model. So in order to train, we need a learning rate, right? So it's, again, we don't have much time to cover the details today, but especially like how much you want to update the weights, right? So for neural network, we have weights for each neurons. So it's like how much you want to update the neurons, the weights. So this learning rate times the gradients, and that's how much we update the weights. So learning rate is basically how much you want to update the weights. And epoch is how many times you want to train the data, right? So one epoch is if we because we train by batches or mini batches, one epoch would be like we run the whole data set once. So how many mini batches it may take, but once we go through all the data, that counts as one epoch. And here we're doing 50 epochs. So we're gonna look at the data 50 times, and then we can train the model. So this may take a while to train, so we just start the training, and then we can look at how we train the model in the code. Now it's training, right? So we're doing 50 epochs. Now it's 3, 4. It takes about like 2 seconds for each epoch. So it's it's quite fast for these types of data. So we can take a quick look at how we train the data. So here, you specify a device, because if you don't have a NVIDIA GPU, then you can just use a GPU. So it doesn't prevent you from using deep learning if you don't have an NVIDIA GPU. So in order to train the data, so we use a for loop to for how many epochs we want, and we set the model into train mode. And we also log the losses, so we can look at whether the model's performance improves over time, right? So here we use an optimizer, which basically we use that to update the weights, and then we run. We have the input. We use a for loop to grab a mini batch from the train data loader, right? So X is the input, Y is the output. In our case, X, Y are the same. So you run the input through the model and you get your output. Then we can calculate the loss, the loss, right? So here we're looking at mean squared error loss, just to calculate the differences between the output and our target, which is the input, right? So we want to minimize the loss. We want the output from the model as make it the same. We cannot do that, but make it as close as possible. So we calculate the loss using the model output and the input, and then we use the loss to update the weights. Then we can this just for plotting purposes, we can look at the losses over each epoch, and then use the validation to calculate the validation loss as well, and then just record the losses. We can also print maybe every 10 epochs, and that's basically the training process. So okay, it's now finished. Now you can see that we print the train loss and the validation loss every 10 epochs. You can we want to look at the validation loss. Like you can see that the loss is going down. It went from 0.62 to about 0.60. So obviously our model is learning, so we want to look at the losses to see how it improves over time. So the blue line is the training loss. As you can see that it goes down. Same for validation loss. and it goes down and it reaches a plateau, like it doesn't improve much over, say, after 40 epochs. So this is good, right? So we want to stop with the training if the validation loss is not improved anymore. So this looks good, and then we can run the. So we finished the training. Then we can run the whole data set through our model, right? Because we want to extract the outputs from the, because we don't care about the output, right? Because it's just the input. We don't care about that. We want the bottleneck layer outputs, right? So in order to do that, we just again build a data set and a data loader for the entire data set without any splitting, and then we get the encodings from the bottleneck layer using this function, and then we can check out the encodings because we use the 20 for the bottleneck layer. So we can imagine that the shape of the encodings should be the number of samples by 20, right? So yeah, as expected, this is our total number of cells, and we have 20 for as columns, which are the 20 neurons that we specified in the model. Just a quick look at the function, the gating encodings function. Yeah, so basically we run the model, but we don't run the entire model, right? We just run the input through the encoder without the decoder, because after training, the decoder is basically useless in our case. So we just run all the data through the encoder and get the encodings, and that's the basic idea. So we got our encodings, right? Now we want to visualize the data, right? So fortunately, we have already labeled the each cell. So we using some like manual process to label each cell with cell types. So in our case, these are immune cells. So we have T cells, like B cells, stuff like that. So we'll see, how our data would look like, right? Do we separate different cell types on the two-dimensional space, right? So we want to check that out. So we load the metadata, which contains the cell type information, and I did some label cleaning. So now, because we have 20 neural dimensions, right? But we want to visualize in a two-dimensional space. So here we need to use another tool called UMAP. UMAP is very commonly used for single cell analysis for visualization. So we would do that. So we use UMAP to encode the 20-dimensional data to a two-dimensional space so that we can visualize them. So by the way, for golden standard single cell analysis, like say if you are using Sera or ScanPy, you first do PCA, right? To reduce the data to say, I don't know, maybe 30 or 20 PCs, and then you use UMAP to visualize the data. Here we basically replaced PCA with our autoencoder, and then use UMAP to visualize the data. So it would be interesting, if you have say you want to do a side project, right? So you do PCA, using Sera or ScanPy, and then use UMAP to visualize the data. And you want to know, whether deep learning can improve this. So you, instead of PCA, you do autoencoders to reduce the data to say a 20-dimensional space, and then do UMAP, see, whether we got a better result. So that would be like a little fun project to do, after this tutorial, this today's lecture. So now we can look at the data. So this will be our final result from the autoencoders, right? So we have different encoders are different cell types. As you can see that they are, our model did a reasonably job, a good job, separating those different cell types, right? So here we have, monocytes, they form a different cluster, and these are progenitor cells, our B cells, CD8 T cells, CD4 T cells, and NK cells. So you can see that, the different cell types are well separated on this UMAP. It shows that, our encoders did a, good job at, dimensionality reduction, right? So it's quite good. So here by comparison, I'm showing you. So here we're looking at UMAP that using the same model but trained on the gene expression data only. We don't have any protein data in this plot. So you can see that like we cannot separate these cells, right? We have green cells on the bottom, CD4 T cells, and we have like these light blue cells on the top, CD8 T cells. They're just mixed together, right? We cannot separate them. And these NK cells are clustered very close to CD8 T cells, right? It's just a mess. And the mid cells is also on top of these other cells. So it's hard to separate those clusters. But by comparison, if we add protein data, now the cell types are much more well separated. It just shows you, like, by looking at RNA data only, you may not get a very comprehensive profile of the cells. Adding protein data really give you extra power at separating different cell types and identify heterogeneity within the cell population. Yeah, so that's my lecture today. So I hope you can check out the resources afterwards if you're interested. Check out the PyTorch, check out the single cell analysis packages like Sera, ScanPy, and SCVI, and look at the code as well if you're interested, and try to implement your own neural networks. And yeah, and thank you for your attention. That's all I have today. Thank you.
So if you have one or two questions you can ask, then we will wrap up session. Only one or two questions. We have only time for one or two questions.
Tell me, sir, I have a question.
Sure.
Sir, I want to ask if I have to apply on my data, so the encoding part, just you say in your second slide. Can you show me the slide, the previous slide?
The slides?
The first part of this script.
You mean the PowerPoint?
No, not the same script to encode to compress the data.
Import the data like here?
To compress the data. When we can compress the data.
Sorry, sorry, I don't quite.
You also showing your slides.
He's talking about compressing the data.
Oh, compressing the data. You mean the model?
Yes, design the model when you compress the data. Mm-hmm. This part is a little bit confusing. Yes, this one, self-encoder. Self-encoder underscore RNA.
Yeah, so if you just for RNA, right? So you said the pro to in features of protein to 0, right? And hidden pro to 0. So for the final encoder, you have both. You compress both to this 20 dimension or 20 neurons. But if you set this to 0, right? You just compress the RNA to 20 neurons. So it would work if you just set like when we initialize the model, we have number of features for protein, right? In your case, you just set this to 0. You just say put 0 here, and hidden RNA how many you want, right? And then hidden protein to 0. So then if you do this, it would just work for our RNA only. Does that make sense?
Okay, a little bit understanding.
Sorry, I have a question.
Sure.
Okay, so is this good for omics integration actually? Like, could I also use it for methylation and stuff like that? Or is it just like, because what you showed is like the compressing of RNA and the compressing of the protein data, but could I also, like, for example, put in that methylation data or stuff like that? Would that be possible? Is it good for omics integration or not? Or is it the way?
Basically anything, you can use it for anything that you can put, if the data is tabular, right? So you have, rows as samples, columns as features. If your data is in that kind of format, you can use it. So it doesn't, the model doesn't care if it's RNA or protein, doesn't care.
And what is the good thing about, like, if I could do the same actually with a PCA. So what is the better thing about what you're doing, and if I would do it with a PCA, what is
Yeah, that's what I was trying to say, that, right? So if you want to save yourself, you can do a little project, right? You can use this and then use PCA and compare. So the idea this one is better is because PCA does linear transformations, right? Everything's linear, but we know that not all the information is linear, right? But for deep learning, because we use those nonlinear activation functions, right? And we have so many layers. And so the idea is that it can better capture the nonlinear information within the data. So that's the idea.
Okay, I got it. Okay, I got it. And like, because for omics integration, like there's Diabolo, for example, which I know, what would be better? Like, because what I know so far, which would be the best way to do it, is actually Diabolo. And is this better, or how would you compare your thing you did with Diabolo?
Yeah, actually, I have never used Diabolo. I've never used it, so I cannot say that with much confidence. But if you want to try, you can just compare the result to see if it works for you. And also, this the example we look at today is very simple, so I don't expect it to perform very well. But I do encourage you to check out SCVI. So it is a much advanced, it has much advanced models and functions. It doesn't use autoencoders. It uses a variation of autoencoders. So when you encode the data, you don't just code into a number. You encode the data in how do you say, you encode the data to get two parameters for distribution. So you basically learn the parameters for say a gamma distribution. So that's called variation autoencoders. You may want to check out SCVI to see if you can get a better result using that approach.
SCVI, okay.
Yeah, SCVI, like a single cell VI. I don't know. I don't remember what VI stands for. Sorry.
Variation integration or something, maybe. Yeah.
Inference. Oh, yeah, the inference. I think that's it.
And one last question, because I didn't get quite well what the target is at the end, because we have these clusters, you build it at the end, and then we have this, but these clusters are the thing that was made by the UMAP, isn't it? Like, is it tested against the real things? Like, because now we have this, but is this, so where is my score? Like, I'm kind of looking for a score, how well it performed.
Yeah. So here we're just looking at, like, how do you say, it's not quantitative, right? But we're looking at how well can we separate those different cell types. We know the cell types, but we don't have this visualization, right? So we basically use the this the final picture was generated by UMAP, but UMAP was run on the outputs from our autoencoders, right? So its business shows that the autoencoders can separate, it's good for visualization purpose for this data. So it shows that it can, when you visualize the data on the two-dimensional space, it can separate those different cell types. So if you want to quantify this, if you want to score, you can use like different metrics, like a silhouette, which shows like how separated those different clusters are. I would check out a silhouette width, and then there is also like normalized mutual information, right? To just to show how these here we're just looking at dimensionality reduction and visualization, but you can also use the 20 dimensional metrics for clustering as well. So if you do clustering, then you can compare the mutual information to look at how your clusters are. So you can use those metrics for scores if you want. So and then you can compare that to PCA. So for example, you do PCA first, and then do the same thing, and then you do autoencoders, and then use the silhouette or normalize the mutual information to compare whether this is actually better than PCA or some other methods.
Thank you very much.
Excuse me, sir.
Sure.
Sir, actually, during PCA, we have to select which PCs we want by seeing the dimensional plots or PC plot. So here you are choosing dimensions for 20. So how can we visualize the dim plots and then decide that how many dimensions to take for our downstream process?
Yeah, so this is the one major difference between PCA and this. So in PCA, the first component is the most important. It counts the most variance among the data. So so you have your 30. The 30. One is the most important. 30 is the least important. But for this, everyone is equal. So it doesn't matter like whether you are the first column or the last column. They're equally equal. So you have to take all of them. You don't select. If you want to use fewer, say 20 is too much. I just want to use 10. Then you set the ZDIM parameter to 10 to compress the data to 10 neurons. So you don't select, you don't compress the data into 20 and then select the 10 of them. You cannot do that for this because every column is equally important. It's not like a PCA.
Sir, but then there is a chance for increased noise in the data.
Increasing, sorry?
Increasing noise in the data. Unwanted data.
You mean noise?
Yes.
Right?
Yes,
Yeah,
noise in the data.
Yeah, so basically, I didn't show the details here, but for our first layer, when we encode the RNA and the protein directly, we have a P parameter, which is the dropout rate. So when we compress the data, we don't read all the information. So 0.1% means that we drop 10% of the input. So that's a way to do denoise, right? So we just we randomly drop 10% of the genes and 10% of the proteins. So that's one way to help us denoise. And the other approach you can do is to reduce the ZDIM, right? So instead of 20, you just use 10. And if you want to further denoise, you can increase the P parameter, and that's things that you can experiment with.
Okay.
Yeah.
Thank you, sir.
Thank you.
Yeah, sure.
So I think we are running over time, and if you have still more questions, you can write to us on email. We will forward your email to Dr. Yuan. So at this point of time, we would like to end this workshop. We also would like to thanks for your all kindness and efforts to participate in this workshop. Also, we would like to thank Dr. Yuan Tian for delivering this amazing lecture on deep learning. We are honored that you deliver on our platform. So thanks, Dr. Yuan. Thanks all participants, and goodbye to all of you with a hope that you learn a lot of things with decodelife.org since last 1 month. So thanks Dr. Yuan. Now I seek your permission to end the session.
Okay, thank you so much for your attention. Good luck, guys.
Thanks.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
So there is one more request. Please fill out the
Thank you, sir.
feedback form so that we can improve ourselves, and also please subscribe our YouTube channel so that you can get amazing videos on bioinformatics. So thanks and goodbye.